Hey guys, and welcome, and today we're going to do Edexcel AS level Biology B exam recap for Carbohydrates 2. Okay, so when lots of monosaccharides join together, they form the polysaccharides, in which the monosaccharides are the monomers and the polysaccharide is the polymer. And the polysaccharides have many sugar units, and they can either be unbranched in amylose, or if it's an amylopectin and glycogen, the chains will be branched. In starch, it will be a mixture of amylopectin and amylose, and that starch is the energy store in plants. Glycogen is a carbohydrate store that's found in animals. Right, alpha glucose monomers. So in starch, the amylose is actually a straight chain, but what happens due to the hydrogen bonding, it will coil and look like this. And that makes it a compact store of glucose and therefore has a lot of energy. In amylopectin, it is branched with 1,6 glycosidic bonds, and so they don't allow much coiling. 1,4 glycosidic bonds are the ones that result in hydrogen bonds that cause the coiling. This means it is more easily broken than amylose, and so it gives the starch a mixture of rapid and slow glucose energy release. Glycogen is like amylopectin, but more frequent branches of 1,6 glycosidic bonds. Therefore, it will look mostly unbranched, like in amylopectin. This means it can be broken down even more easily than amylopectin, releasing the glucose energy even faster, and that's needed in animals. Making and breaking polysaccharides. So saccharides are joined in condensation reactions which form glycosidic bonds to make the polysaccharides. In alpha glucose, the monomers are alpha 1,4 glycosidic bonds in amylose, and alpha glucose monomers with alpha 1,4 and alpha 1,6 glycosidic bonds are in amylopectin and in glycogen. In beta glucose monomers, they will form beta 1,4 glycosidic bonds, and they will have the monomers will alternate, so they will flip. So actually, it is flipped over each one, and that's in cellulose. And when starch, glycogen, and cellulose are broken down in digestion, the enzymes will catalyze the reaction by a hydrolysis reaction, which will input with water to break the bonds. Beta glucose, as in polymers for cellulose. In cellulose, the chains do not coil, but straight lines next to each other. The alternating unit inversion allows hydrogen bonds between the hydroxyl groups in one area and the oxygen atoms in the other molecule. These hydrogen bonds then hold the chains together to form strong microfibrils. As you can see here, it is inverted, and we have the hydrogen bonds in between the straight chains. Now, distinguish between the structures of amylose and amylopectin, where we said amylose is straight chained, whereas amylopectin is branched. Amylose has 1,4 glycosidic bonds only, whereas amylopectin has both 1,4 and 1,6. Okay, now try this. Which of these statements of cellulose are true? And you can select more than one. So it is a polymer of glucose. Yes, it is a polymer of beta glucose. It contains alpha and beta glucose. No, it only contains beta glucose. It has glycosidic bonds. Yes. It has side branches. No. Finally, a molecule of it can form hydrogen bonds with adjacent molecules. Yes, that is correct. So A and A and C and E are the correct answers. And we are done.